Okay, so today we will be discussing about the metabolic pathways that are used mostly found in industrially relevant organisms. So these pathways, we will not be delving too much on glycolytic pathway as much as we will be looking at other pathways aside from glycolysis and aerobic respiration. So let's look at first the two types of metabolic pathways. You have the catabolism and anabolism. Catabolism is an energy generating pathway. So you degrade large molecules into smaller molecules, whereas in anabolism, it's an energy utilizing pathway. So you use energy to create, to synthesize. It's biosynthetic. So you to synthesize large molecules from smaller ones. So to give an example, your catabolism, an example of catabolism is aerobic respiration, whereas anabolism is DNA synthesis, is an anabolic pathway. So this is one way to look at it. So most of the time, you have the food molecules as the reactants in catabolic pathways. And you use the energy from catabolic pathways to actually create we use the energy from catabolic pathways to fuel the anabolic pathways. So that is why inside the cell, you have the two pathways occurring simultaneously. So they use catabolism to fuel anabolism. So for catabolic pathways, it's the breakdown mechanism. So you have here, example of that is the substrate level phosphorylation. So you create energy by breaking down your energy, or rather breaking down molecules. So mostly the products here is ATP, NADH, NADPH, FADH2, and of course heat, as there's always an excess heat generated. So examples of catabolic pathway, aside from glycolysis, you have the this is actually glycolysis. Embden-Meyerhof-Parnas pathway is the most common route found in most organisms. So it occurs in both aerobic and anaerobic conditions. And you have the three regulatory key enzymes. We already discussed this EMP pathway. This is glycolysis. This is the 10 reactions of glycolysis. The alternative name is the Embden-Meyerhof-Parnas pathway. This is actually an older term. If you look at older books, it's called Embden-Meyerhof pathway, or the EMP, because you have the third person Parnas. This is named after the ones that discovered this pathway, Embden-Meyerhof, and of course Parnas. Now, we say that we have the three key regulatory enzymes. So this is a tightly controlled pathway. Just like other metabolic pathways, there must have been control. Now, the three regulatory enzymes in glycolysis is the hexokinase, which is the first step, where you have the glucose, you add ATP, or rather you add a phosphate to glucose. The second regulatory step, and the most important one, is the phosphofructokinase, which designates this particular molecule to the glycolytic pathway. So this one adds the second phosphate to fructose 6-phosphate. The third one is which is the third enzyme. So the third one is the pyruvate kinase at the bottom, the one at the bottom. So that's the third regulatory step. So that this pyruvate kinase is the one that produces pyruvate, and also the energy harvesting mechanism for glycolysis, or rather for this EMP pathway. So why does it have to be tightly regulated for the last step? It's because ATP is quite a reactive molecule. So if there's an excess of ATP in the cells does not need to produce more ATP. So it's one of the tightly regulated steps. So this is your glycolysis, or the Embden-Meyerhof-Parnas. So aside from that, you have the pentose phosphate pathway, PPP. So you have another name is the hexose monophosphate pathway. So this operates with the EMP pathway simultaneously. Like the EMP pathway, it can also be done, or rather it runs in either aerobic or anaerobic conditions, and it's anabolic. So what's the difference here? So if you have here it's mostly cyclical anyway. So you have 3-glucose-6-phosphate. So it starts also with, it shuttles off 3-glucose-6-phosphate from the EMP pathway, and it is used to produce your, uh, the rather, the metabolites here are used, the intermediates in these pathways. For example, 3-ribulose, they are used to synthesize other molecules for the cells. For example, the we can shuttle ribulose phosphates here to create nucleotides. So this is mostly, it shuffles. This pathway shuffles the carbons of your, this shuffles the carbons. You can actually produce here the ribose 5-phosphates, glyceraldehyde 3-phosphates. So these are the important intermediates. This is just the intermediates of the pathway, but these intermediates are important because they can be used as starting materials to create other molecules such as amino acids, nucleic acids, and other important biomolecules. Even the electron carriers, NAD plus, NADPH, these, they can be used or they can be generated using this, pentose phosphate pathway. Then we have the Entner-Doudoroff pathway. So these are, used by microbes that do not have the mechanism of the EMP pathway. So if they do not have the EMP pathway, this is the alternative. So these are found mostly in the gram-negative bacteria, which you have Azotobacter, Pseudomonas, Rhizobium, Xanthomonas, and Zymomonas. Now, in this pathway, you start off like, glycolysis. You start off with glucose, and end up with pyruvate. But here, the difference here is you have your phosphogluconate. Instead of, adding additional ATP, because you have here EMP pathway, remember you have 2 ATP investment steps. Here you just have 1 ATP investment step. And then you will produce 2 molecules of pyruvate. So they share commonality in such that you have your, this second part, the lower section of this ENT pathway is, or rather is shared with your EMP pathway. It's actually quite similar, but the first stage is different. So here, it just produce one ATP, one net ATP gain for your pathway. So for the phosphoketolase pathway, it's also called the Warburg-Dickens pathway. So this one happens in lactic acid bacteria. And this pathway is used for heterolactic fermentation. When I say heterolactic fermentation, that means this fermentation produces lactic acid among other fermentation products, such as, in this case, ethanol. So this pathway, the PK pathway, is a fermentation pathway, whereas starting off with glucose as well, but ends up with the products lactate and ethanol. Instead of pyruvate, you produce ethanol and lactate. And that's why it's called heterolactic. Hetero, it's different. So it produces 2 different products. One of them is lactate, the other is ethanol. So unlike the lactic acid fermentation, where the product is just lactate, here you produce both lactate and ethanol. So that's one of the main or key differences here. Now, continuing on, you have the TCA cycle. This is your citric acid cycle. So this happens after the glycolytic step for normal oxidative respiration. So this one is used to catabolize pyruvate. We have already discussed it here in a separate video. So I won't be looking at it here. Now, the other one is the glyoxylate pathway. So glyoxylate is just like your citric acid cycle, but it's a bit different. Whereas, although you will see commonalities here, you started with acetyl-CoA, you have oxaloacetate as the receiver to form the citrate. But unlike your pathway, this one produces succinate to acetyl-CoA, NADH, and 2 hydrogen molecules. So it's a different version of your citric acid cycle, and one of the key metabolites here is the glyoxylate. That's why it's called glyoxylate pathway. Glyoxylate can be used as starting materials for other, or metabolites for other pathways, other metabolic processes. Okay, so looking at this pathway, we can say that respiration can both be aerobic or anaerobic. So for microorganisms, we have aerobic and anaerobic respiration. Aerobic respiration is the oxidative type of phosphorylation, and you have your chemoosmotic process here. But anaerobic respiration, it also has an electron transport system, but you have having oxygen here actually disrupts this respiration. So these steps here, these are substrate level basically phosphorylation, or rather the energy here is through substrate level phosphorylation. You produce ATP here, but through shuttling the phosphates from another molecule to ADP. Now, for the aerobic step, the oxidative phosphorylation step, it can be divided into both aerobic and anaerobic. For the aerobic, we have the electron transport system, whereas you have your oxygen as the final electron acceptor. But for the anaerobic respiration, you also have the electron transport system, but you do not have oxygen here. So let's look at the difference. So in the normal aerobic respiration, you have your electron transport system. You produced water as the end result. And oxygen is the final electron acceptor, as you can see. But for the anaerobic ones, you have a different molecule as the final electron acceptor. But again, the system is very, very similar. In the case of the nitrogen fixation, you have NO3 as the final electron acceptor. You have the sulfates for sulfur bacteria, and even for methane bacteria or methanogenic bacteria. Carbon dioxide is the final acceptor, and they produce methane. So they both utilize an electron transport system. What is different here for the anaerobic organisms is that they have a different electron acceptor. So for aerobic respiration or aerobic organisms, oxygen is the electron acceptor. For the anaerobics, they utilize nitrates, sulfates, or even carbon dioxide. So nitrate respiration, they are found in facultative anaerobes. So when we say facultative anaerobes, they can survive in the presence of oxygen, but they do not really require oxygen. So they have the redox potential. The reason why some organisms, or rather most organisms such as us, most of the larger eukaryotes are aerobic in nature. They perform aerobic respiration because aerobic respiration produces a larger amount of energy, because the redox potential of oxygen is higher at 0.82 versus for the facultative anaerobes, which is 0.42. So almost less than 50% increase. Now, but some organisms still tend to do nitrate respiration because they really don't need the very large amount of energy that they will get if they use oxygen as the electron acceptor. So for the nitrifying bacteria, so this is the reduction of your nitrates, because for nitrate respiration, nitrate is the final electron acceptor. So you reduce the nitrate, and it's called the nitrification. So examples of organisms that do this is the Pseudomonas, Paracoccus denitrificans, and Thiobacillus denitrificans. So they use nitrates. And that actually helps, as denitrification is important steps in the nitrogen cycle. Now here is your anaerobic respiration that utilizes your nitrates. So this is just a simple pathway. So in the process, you produce nitrogen gas. So aside from that, you also have the sulfur respirations. So most of them are your sulfur producing bacteria, the sulfovibrious species. So these are usually found in obligate anaerobes. So when we say obligate anaerobes, if you expose them to oxygen, they die off. So that is because oxygen disrupts their pathway, specifically this particular pathway. The fixation of instead of sulfate acting as the final electron acceptor that should be for this sulfur bacteria, oxygen interferes with the pathway, and since oxygen has a larger redox potential, they produce a much larger amount of energy that they cannot handle, so they basically die off. Other examples are the carbonate respirations. So they are found in the archaea bacteria. So they are the methanogens, Methanococcus and Methanobacterium. So they utilize carbon dioxide as the final electron acceptor, and in the process they produce methane. So additional fermentation steps. So we had already discussed in in a different video the the alcoholic and the homolactic fermentation steps. For the alcoholic fermentation, you recycle the reason why we do fermentation, just remember, it's to recycle the electron carriers. And we use pyruvate as the starting material here, which is the direct product of your glycolysis. And for alcoholic fermentation, we produce carbon dioxide and ethanol. That is why beer products are carbonated, because that's one of the byproducts of the fermentation of your Saccharomyces cerevisiae. Now, for the lactic acid fermentation, our cells also do homolactic fermentation if there is not enough oxygen available. But we produce lactate, and lactate is quite dangerous if there are too large amount, because of course this is acidic. So this is also found in the lactobacillus. Well, example, if production of yogurt. So they produce lactate. This is homolactic fermentation. And then the other one, this is the PK pathway, the heterolactic fermentation pathway. They utilize the phosphatidylase pathway to produce lactate and ethanol as the byproducts. So aside from those, we also have other fermentation pathways. Mixed acid fermentation, whereas you produce lactate, ethanol, acetate, carbon dioxide, and hydrogen gas. So depending on the activated pathway. So you can also have butanediol fermentation. The end product is butanediol. And propionic acid fermentation, where the end product is propionate. So different organisms utilize each their own pathway, and because of the end products of these different pathways, so here propionate, and then the other one is the butyric acid fermentation. This one, isopropanol, butyrate, and butanol are the end products. So because of the end products of this fermentation pathway, we can use them to actually identify the microorganisms, to allow us to characterize what type. For example, you have a culture of unknown organisms. You want to characterize them. Is this a novel organism? Am I the one that discovered it, or has this one been discovered already and I'm just seeing this manifestation here? So the one of before the sequencing era, the genomic sequencing era, the only way to do that is looking at the metabolic pathways, the fermentation products. So you culture them and you study how they behave, their metabolic activities. Do they produce butanol? Do they produce propionate? Do they produce ethanol? Do they produce lactic acid? So those are the earlier, the old school way of characterizing microorganisms. Although we are still doing that. But for identification purposes, recently we are just getting the DNA of them, their DNA, and then sequencing them, and then looking at the database. Because most organisms, some organisms, they adapt to different fermentation pathway. So some organisms, they do not only do homolactic fermentation. Under different circumstances, they can do heterolactic fermentation. So that's the one thing about the microorganisms. They are highly adaptable. So anyway, that concludes our lecture about the other metabolic pathways, or rather most metabolic pathways in microbial organisms.